on? Can you hear me?
Yes, very fine.
Thank you. I'm sharing my presentation.
Would you please start the session giving us an introduction about the international webinar, this webinar, and then we can go to the lecturers and go ahead with the webinar. Thank you.
Okay, we started.
Yes, I guess it's okay to start. We are just on time.
Thank you. In the name of God, hello dear speakers and participants. Today, 60th of November 2022, we start a conference, International Microbiology webinar, with the emphasis on Probiotic. The webinar, organized by Iranian Society of Microbiology, Tehran University of Medical Science, and Iran Dairy Industry, PEGA company. The member of panel, Dr. Saeed Mirdamadi, Dr. Farzaneh Aziz Mohseni, Dr. Mardane Furubi. International invited speaker, Dr. Huling Fu, Dorothy Eskesen. Panel time 130 minutes, and the question and answer after each presentation 5 minutes. We hope to have a scientific and valuable seminar. We have 5 presentations, and each lecture lasts 20 minutes. During the lecture, you can type your question in the box chat, and at the end of the presentation, we will answer to your question. And let's start with the first speakers. Dr. Huling Fu. Dr. Huling Fu, let me introduce of Dr. Huling Fu. Dr. Huling Fu, BSS Homes, UPM, PhD from London, and postgraduate certification in education, Department of Bioprocess Technology, Faculty of Biotechnology and Biopolymers Science, University of Putra, Malaysia. Dear Huling Fu, can you hear me? Are you connected?
Yes, I can hear you clearly. Can you hear me?
Yes, very clearly. Dear Dr. Fu, I hope you are good, and thank you for participation in this webinar. And you have 20 minutes for your presentation. Please start your presentation. Thank you.
Thank you very much for your kind introduction. Okay. I'm so sorry, this is not the one. Wait. Okay, good morning everyone again. First of all, I would like to thank the organizing committee for their kind invitation for me to share some of the results that I obtained with my research team member. Okay, and it is my great pleasure actually to give some brief introduction of some of the work that we done with my research team until today. So the title for my presentation is The Wonders of Postbiotic, derived from a lactic acid bacteria isolated from nation foods. Okay, all the while, I think you heard about probiotic, prebiotic pretty much. But in my research activity that I have in my research team, we focus on the postbiotic. So what it is that? Okay, so this slide shows you the timeline of the research activity that related with lactic acid bacteria. I think all of you know very well probiotic, prebiotic. Now, within the last 5 or 10 years, actually we have encountered another terminology that has been coined or related with lactic acid bacteria. They are paraprobiotic postbiotic and cytobiotic. So this is what I'm going to discuss with you all or share with you all, mainly on the postbiotic and cytobiotic. Okay, I think application of lactic acid bacteria or LAB as probiotic, I think we know very well. All right, it has been used very extensively in many industry, especially the pharmaceutical, food, and as well the feed industry. And also probiotic culture is not multitasking. I think as we know now, not all single probiotic culture can fulfill all the functionality or benefit features that required by the consumer. Therefore, actually the increasing demand of new probiotic culture that specific to each functionality is under high demand at the moment, especially under the COVID-19 pandemic condition. Okay, what are the challenges of using probiotic while versatile? I think we know very well. We always heard about inconsistent result of probiotic on various consumer or different consumers. And it is because mainly the different viability of probiotic species in product or feed ingredient. All right. And the second point contribute to these inconsistent results is the different colonization pattern and persistent in persistent of the gut, yeah, in the gut that exhibited by different strain of probiotic cell. I think this is not new to all of us. And another point, factors contributing to the challenges of using the viable probiotic cell is high maintenance cost to ensure viability, especially when you deal with the storage and handling process, which is required, always required, the low temperature condition. All right. And following point, I think we also heard some incident about of using viable cell, which is they may induce pro-inflammatory responses in hosts with inflamed gut, especially those have a very bad leaky gut. All right. And another very alarming, report on the immune compromised patient, the high risk of a probiotic can happen to the immunocompromised patient. I will share with you some of the reports that published last year. And the last point is the acquisition of virus gene from pathogenic bacteria through horizontal gene transfer. And this is the antibiotic resistant issue that actually striking throughout the world since last 10 years. Okay, this slide shows you some of the results that we have obtained with the culture of LAB that we isolated from the chicken feces in Klang Valley of Malaysia. And it is quite alarming, all right? The LAB that we cultured from the gut of the chicken or feces, actually they show a lot of antimicrobial resistant profile against the antibiotic that used currently in treatment, also in the feed additive, as a feed additive, such as clamidins, chloramphenicol, amoxicillin, as well as the very common use penicillins, as well as erythromycin, and also the ampicillin. So, and as well as vancomycin. So it looks like they are no longer free from antibiotic resistance now. So we have published this work in these articles. If you wish to know more, you can dig out the article and read up yourself, okay? And another alarming issue is here, sorry. Okay, beware probiotic in ICU patient. This article actually published last year, all right, by Nature Medicine. It's quite alarming actually. When I read that one, I got shocked. And it shows that the probiotic become pathogenic and they can invade the barrier of gut and reach the bloodstream of the ICU patient and cause a lot of problem to the ICU patient and also fatal effect as well. So you can dig out the report and read up for yourself, yeah? Since we don't have much time to discuss on that matter. Therefore, based on the challenges of probiotic that I shared with you just now, my research team focused 100% since year 2003 that we work on the metabolites produced by probiotic, known as postbiotic nowadays. Okay, so what is postbiotic? I think probiotic everybody know. It deals with the live microorganism that confers beneficial effect with a certain amount. Okay, but what is postbiotic? Postbiotic is non-viable soluble factors that contains bioactive metabolites produced by probiotic LAB that mediates beneficial probiotic effects. And these are some of the examples that can present in the postbiotic. And the composition of postbiotic actually depends on the fermentation process that you use for the LAB to produce postbiotic. So they may contain short-chain fatty acid, they may contain extracellular and secreted proteins such as bacteriocin and other short metabolites of bioactive peptides, or free amino acid or dipeptide, depends on the enzyme system that they have within the LAB that you use. And they may also contain some exoplurisaccharides. Okay, I think some LAB can produce some of the slimy compound. They are exoplurisaccharides actually. And they may have contained teichoic acid and also other cell wall component if, let's say, the postbiotic produced with a long processing of the fermentation. Okay, my research team, we work on very closely on 6 strains of Lactobacillus plantarum. Nowadays, according to the new system to name the Lactobacillus genus, it has been known as a Lactic Bacillus plantarum nowadays. All right, there's one publication published earlier this year. They have found out actually Lactobacillus plantarum has different lineage in terms of genetic composition, and they suggest that Lactobacillus to become the Lactobacillus plantarum nowadays. And we work on this strain very closely, and those strains are were isolated from Malaysian foods. Okay, this diagram shows you the summary of the process that we have developed for the postbiotic. So we isolate from the culture from the Malaysian foods. We go through the fermentation process, and we go through fermentation process by using bioreactor, and we spray dry to become the powder form or can be liquid form. And 2 types of postbiotic has been verified for their beneficial effect through various animal models such as poultry, rats, pigs, the fish, tilapia fish, and lately the lamb. So we have been using many animal models to verify their beneficial effect or functionality. And I summarize those functionality we've done for our postbiotic. We try to study the health promoting effects on rats and also livestock animal models like I showed you just now. We also try to study their inhibitory activities against various pathogens or food spoilage microorganisms. We also try to study the anti-cancer effect based on cytotoxicity effect using various human cancer cell models. So this slide summarizes all the functionality that we have obtained for our postbiotic. However, I could not share all of them since because due to the limitation of time, I will select some of them to share with you all today. Okay, so antimicrobial activities of postbiotic, right? This slide shows you the simplified actually for the various combination of metabolites that we derive from the 6 strains of Lactobacillus plantarum that I mentioned just now. So we try to study their inhibitory activity against the Salmonella typhimurium, Escherichia coli, Listeria monocytogenes, and vancomycin-resistant Enterococcus. And it shows all of them, actually those pathogens can be inhibited by various combination of metabolites with different strength. Okay, so the inhibitory actually is quite broad, and the inhibitory activity such as, for example, that's shown by this, inhibitory agar-well diffusion assay with the clear inhibitory zones, we can get those inhibitory against this list of the microorganism or pathogens or food spoilage, bacteria. And it shows that they have a broad inhibitory activity for our postbiotic that we have in hand. And what contributing to the broad inhibitory activity of postbiotic, most likely they have the organic acid. In our postbiotic, 6 strains of postbiotic, they do contain the lactic acid, acetic acid, and also formic acid. And they are heterofermentative LAB in this case. So, as we know that organic acid has been shown to have various health benefits. For example, stimulation of mucus production in the gut. They also can repair the tissue in the gut, and they also induce the IgA production or immunoglobulin A production, and they also promote the Treg cells as well as inhibition of inflammation of necrotype, the factors that can contribute to the inflammation activity. So all of them, the organic acid actually contributing to the gut health condition. And bacteriocin, I'm not sure whether you all know bacteriocin or not. Bacteriocin basically is a group of the short peptides that produced by certain LAB. Not all LAB can produce bacteriocin. And if you can have one culture produce bacteriocin, it's something like you got the lucky draw. Very well, good. So bacteriocin actually are the specific cell protection molecule that against its own against various pathogens. And bacteriocin produced by LAB will not have any impact on the producer cell because they have specific immunity protein to protect themselves from their bacteriocin. And it is a group of ribosomal synthesized antimicrobial peptides or proteins that produced by various genera of bacteria. And normally it contains 30 to 60 amino acids within a bacteriocin molecule. At the moment, to my knowledge, 4 classes of bacteriocin has been classified. All right, it can be from simple up to very complicated structure of the bacteriocin molecule. And it has inhibitory towards many undesired bacteria, including foodborne pathogens, but it depends on the types of bacteriocin. Some of them can be very narrow range of inhibitory, or they can have broad. It depends on the bacteriocin molecule that produced by LAB. And these bacteriocin molecule, not like the antibiotic, they are not resistant and can be degraded mostly by digestive enzyme. And this advantage against classical antibiotic used as for the treatment. And in our culture, the 6 strains of Angel's culture of LAB, they possess 2 classes of bacteriocin, which are the class 1 plantidricin W and class 2 plantidricin EF. And this is the first report that regarding the occurrence of 2 classes of bacteriocin within the same cell. And we have published it in year 2010 in this journal. You can try to read up further if you want to know more about it. And we have had time to actually classify or identify these closely related 6 strains of the Lactobacillus plantarum because they are the same species. They are the only different strain. But in terms of genetic, they are very similar in terms, especially the 16S rDNA. We cannot differentiate them. We use several molecular tools to differentiate them. They are different individuals. And that kind of work published in this journal. Yeah, Food Technology and Biotechnology in year 2010. And we also try to find out whether these 2 groups of the bacteriocin, or 2 classes of bacteriocin, are they different from those reported structure. And we have done some of the, sequencing on the genetic makeup of these 2 classes of the structure gene of the bacteriocin. And this work has been published in Microbiology Factory in year 2015. And in our work, it shows that plant W had a similar 100% identical structure with the reported plantidricin W. But for plantidricin EF, they had slightly different structural gene compared to those published, plantidricin EF structure. And the major differences is at this end of the structure and this part of the structure. So it shows that they are different compared to those reported structure gene of plantidricin EF. Okay, so the next slide, so subsequent data I would like to share with you. some of the, result that can show they can improve the gut health through the restoration and maintenance of gut microbiome homeostasis. And we know that gut microbiome is very important. I think there's a lot of microbiome data that available nowadays, and try to relate various functionality or various health impacts that related to various group of microbiome in the gut. And as we know that gut microbiome can produce antimicrobial substances such as bacteriocin, normally if they come from the probiotic, it will be that indigenous in the gut. Okay. And also the metabolic end product can have some other functionality as well. This is very, very closely related to the functionality of the gut microbiome with the health impact on the host. And they also can produce essential vitamins, as we know that vitamin B, especially, and K. And they also can degrade non-digestible food components such as prebiotic. All right. And this slide shows, of the, this slide shows you the intestinal histology of broiler chickens that fed with different postbiotic under heat stress. We try to see whether the postbiotic can release the stress or not. So we try to use the chicken as a model, and we impose some of the heat stress to the chicken. We try to feed with the postbiotic and see how's the response in our study. And it shows that those heat stress chicken, when we feed with the different types of postbiotic RI11, RS5, UL4, compared to ascorbic acid and oxytetracycline antibiotic fat, and NC is a control without anything. And it shows that our postbiotic RI11, RS5, and also UL4 can have a better villi heights in duodenum, jejunum, and also ileum compared to those ascorbic acid, oxytetracycline, and also control chicken. And same thing happened to the crib depth as well. All right, they have a better crib depth compared to others. The depth is, not as low as the control group, but they are much better than the positive control group as well as ascorbic acid fat chicken. And the villi height crib depth ratio also they improve a lot. So it means that actually overall the postbiotic improve the gut structure, especially villi height and crib depth. Okay, the next one. Okay. So this one shows you the results of the plasma antioxidant activity of broilers that fed with postbiotic under the heat stress condition. Again, all right, it shows that our postbiotic actually, T4, T5, and T6 are those fed with the postbiotic, and T1 is the native control, T2 is the antibiotic with oxytetracycline, and 3 fed with the ascorbic acid or vitamin C. Again, here it shows that the antioxidant profile in the chicken are much better, especially those fed with the RI11 postbiotic compared to control group. All right. Okay. How about immunoglobulin? How about the immune response of the chicken? Again, you can see here actually, those chicken fed with the postbiotic, they have better IgG, IgA, as well as IgM, the green color, compared to these 3 control groups. Okay, how about blood cholesterol reduction potential? What can they do? So this slide shows you the in vitro assimilation of cholesterol by our 6 strains of the culture that we tested. And they can all degrade cholesterol very well, extracellularly or in vitro. And how about compared with the probiotic? They also have the ability to degrade cholesterol after the fed to the rats. But EL4, they do not have any degradation of the, they cannot reduce the serum cholesterol easily. But RI11 is a good, reduction of the cholesterol in the bloodstream of the rats here. So how about postbiotic fat rats? Postbiotic fat rats, you can see that the reduction of cholesterol are significantly different or reduced quite significantly compared to the control. All of them can have a better reduction rate. especially the RS5 and RI11 as well as RG11 postbiotic. So this slide summarizes that direct fat postbiotic can reduce the enterobacterial count in intestine while maintaining the intestinal LAB population, and significant higher crypt depth of small intestine and cecums obtained. And the postbiotic RS5, RI11, and RG11 have significantly reduced the serum cholesterol level of rats by 21.9%, 15.6%, and 15.2% respectively. As compared to direct fat probiotic, they do have the same effect, but have different, percentage in terms of a reduction in the serum cholesterol. So it means that postbiotic can do the same wonders like the probiotic lifestyle. Okay, so this is another chicken trials that we have for the chicken under the heat stress condition. And it shows also that plasma in the blood of the chicken that fed with the postbiotic, they have lower total cholesterol level, they have lower TAG or triglyceride, LDL, and HDL. They have higher HDL but lower LDL, and the ratio of the HDL and LDL as the VLDL as well. So it shows that they have better lipid profile compared to those control group. So these are the journals or articles that we publish about in the rats. You can try to dig up if you want to know more about the effect of the postbiotic. And this another publication that related to various types of the effect. You can try to dig out as well, because I don't have time to dig out, to share with you in detail all of them. And do I still have time? Hello? Can I still continue? Okay, if I
yes, dear. Yes, dear.
All right. Okay. All right. So now I'm going to share the effect of cytotoxicity of postbiotic on various cancer cells, and we can try to target postbiotic as an adjunctive for cancer treatment. So this postbiotic, we name it specifically as cytobiotic, and we try to define the cytobiotic as a postbiotic that exhibits cytotoxicity effect on human cancer cells, well, apoptotic mechanism, and can be developed as natural chemopreventive agent for overall health maintenance, cancer prevention, and also as an adjunctive therapy for cancer treatment. And this is the definition that developed in my master's student, 2016. And this figure shows you the cytotoxicity effect of 6 types of postbiotic against various cancer cells. And MCF7 is a human breast adenocarcinoma cell. HeLa is a human cervical adenocarcinoma cells. HepG2, human liver adenocarcinoma cell. HC29, human colorectal cancer cell. And K562, HL60 are the human chronic leukemia cells. All right. And the other two is a normal human breast cell as a control, and the other one is peripheral blood mononuclear cell also as a control. So in these figures, it shows that different postbiotic, they have different cytotoxicity impact on different cancer cell line. And you can see that very clearly, they have very, very significant cytotoxicity effect on the human chronic leukemia cell, especially non-anchorage cancer cell. But they do have some different extent of cytotoxicity against anchorage cancer cell. For example, the breast cancer cell, HeLa cell, HepG2 cell, HC29 cell. So it looks like for the HT29 colorectal cell only, 2 postbiotic have a more significant cytotoxicity effect compared to others. But for the UL4 postbiotic, they have very good effect on the MCF7 cell. And this is what we have focused on for our subsequent study. And against the normal blood cell, they do not have any significant cytotoxicity impact. It shows that they are safe to be used as a treatment in the future. Okay, so this is a study that we try to use various blood that we draw from the rabbits, humans, dogs, and chicken, and we try to see whether they have the, osmotic pressure or have the hemolysis activity or not. It shows that they are very safe. Very, very minimal activity that of the, hemolysis occur to the various types of the blood cell. And we use 1% of the SAS as a control and water as a control. It shows that they have more hemolysis activity compared to postbiotic that we have tested on. Okay, so this slide shows you the proliferation of the, breast cancer cell by using 15% of the, postbiotic and 30% of postbiotic. And the cytotoxicity activity actually depending on the incubation time and also the dosage of the postbiotic used here. And the next slide is work on the HC29 cell. It looks like the cytotoxicity activity is not as great as a breast cancer cell just now, but it still has a certain extent of the cytotoxicity activity against, that at different concentration of 50% and 30% of concentration. So at the 72 hours, actually, you can see that this blue bar that produced by the TL1, sorry, not TL1, the RG14 postbiotic can have a very good inhibition on the HC29 cell here. Okay, we also try to determine whether the cytotoxicity is due to the apoptosis or necrosis. I think we all know that the apoptosis is the most preference way to kill the cell within the cancer patient if possible. So we try to differentiate or try to identify the cytotoxicity is due to the necrosis or due to apoptotic. So we use fluorescent dye to study the apoptotic mechanism of the postbiotic that induced on the breast cancer cell here. So it looks like 24 hours, 48 hours, 72 hours, you can see more apoptotic cell that present within the 72 hours, which is shown by these cartoon signs of the, it looks like the Mickey Mouse face. You can see the apoptotic body is present within the cell and the cell become blabbing. And this is shown of the, apoptosis of the, cancer cell, especially. You can see that apoptotic body is, producing or coming out from the cell that go through the apoptotic mechanism, all right? And this is a control cell. They grow healthily, from 22 hours to 72 hours. Nothing happened with the cell that we use for our study. Okay, we also try to use another method to verify the apoptosis mechanism that induced by the postbiotic. We use an enzyme 5 and PI staining method and then detected by the flow cytometry. And it shows that for the untreated cancer cell, breast cancer cell, there's not much of the apoptosis that happen from 24 hours up to 72 hours. They live happily, actually, not much. Cell become the apoptotic. And for this slide, it shows that those, cancer cell treated with 15% and 30% of the postbiotic, UL4, you can see that about 48% of the cell enter early apoptotic after 48 hours of incubation, and up to 71% of the cell enter late apoptotic, phase or late or necrotic phase after 72 hours of incubation. Okay, we have published those work in these two subsequent articles. You can dig out to read up more and come to conclusion. Maybe you can answer yourself now. Probiotic health effects of lactic acid bacteria depend on lice or Weber cells or their metabolites. So in our study, we have shown very conclusively or clearly that we do not need Weber cell of probiotic to have beneficial effect, but they rely on the metabolites produced by probiotic cell. Okay, so this is the research team that I have by students and also my team member. Prof. Low is the one that closely related with my research team that who has verified the functionality of the postbiotic for me all this while. So these are some of the pictures of a UPM, University Putra Malaysia, that I work until today. All right, so that's all. Thank you for your attention. I hope that I do not over the time too much. Yeah, thanks a lot for your attention and patience.
Thank you, dear Dr. Fu, for your excellent presentation. We took advantage from your good presentation, and thank you for you. If every participant have a question, can write your question in the chat, and dear Dr. Fu answer your question.
Or maybe they can ask me a question through orally also can.
Yes. Thank you, Dr. Dear Fu. It seems that nobody has questions, and we start from the next speakers. Thank you for your presentation.
Yeah.
Thank you. Second speaker, Dorothy Eskesen, MSc Human Nutrition Senior Scientific Manager, Food Culture and Enzyme, Dairy Health from Denmark. Dear Eskesen, are you connected? Are you hear me?
I can hear you, yes.
Yes, thank you. Dear Dorothy Eskesen, I hope you are well. Thank you for your participation in this seminar and in this webinar, and I hope you are well. Please present your article. Also, you have 20 minutes time, maybe 5 minutes more for answering a question. Please start your presentation. Thank you.
Thank you for the introduction. It'll be a pleasure for me to continue the talk then on the live probiotics and focus on the relation to the immune system. And of course, I would like to thank the organizers for inviting me to this great series. I will focus on two lactobacillus strains. It's the LTG and the LK 431, where we have a lot of science behind or related also to the immune system. But I don't think we nowadays can talk about benefits of probiotics without also touching upon the human microbiome. So that I will just share a few slides on. But first, again, to repeat the definition, it was also mentioned in the previous presentation that it is these live microorganisms that when administered in adequate amounts confer health benefit. And that is a definition by WHO and also agreed with the International Probiotic Association. What I would like to stress, though, is that probiotic effects are strain specific. So that means that if we have data on a strain belonging to, let's say, a bifidobacterium species, you can't just extrapolate that data and the results to any strains that belong to that species. So we really need to prove strain by strain the evidence. So in human clinical trials, if the probiotics are going to humans, we need to set up randomized control trials to really show the benefits. So that is one thing about the science, but then also, of course, comes to having then the benefits to go out to the consumer. And this is then the responsibility of those marketeers to ensure that they actually do set out true probiotic products, living then up to the requirements in the definition, meaning that the bacteria should be in the product live throughout the entire shelf life of the product. The amounts, of course, should then be linked to the science that is behind and the benefits that we would like the consumer to feel. So if the documentation is on a daily intake of 1 billion per day, then we should ensure that consumers can actually achieve it in a daily serving. And that, of course, can be if it's a dairy product in 100 grams or in a 200 gram daily serving, but we just have to make sure that we actually live what we have, live what we say, so there is science behind. The health benefits for probiotics so far have focused on gastrointestinal health and immune benefits, but of course with new insights on the microbiome, we are seeing new areas coming along, and I think the future is really promising for also entering new health areas and also new target groups. Some may come to food products and some may more take a medicinal route. History of probiotics, I think, is also very clear to all that this has been around for many years now. The first idea is really coined by Metnikov more than 100 years ago, indicating that there would be toxic producing bacteria in the gut that was sort of really impacting our health, but that it should also be possible then to replace these bad bacteria, you can say, with good bacteria from fermented dairy products. But of course, it took years before we really saw products coming to the market. Now in recent years, I would say we have really seen a revitalization of probiotics, and we believe that it is really the explosion in microbiome research that has taken us to new levels, and also interest and knowledge by the consumers. And just looking also on the science, if you Google in PubMed and type probiotics, you can see the orange bars. This has been a steady output on publications. But if you look then into, if you type gut microbiota or gut microbiome, you can see a complete different curve. It's more exponential. So a huge amount of publications are coming out in this field, and it really does hold promise for future solutions, but also for current solution, because the more we understand of the microbiome, also on the strains that we use today, the more specific we can be, both in terms of people that we would like to target and also on which health indications. We do know a lot more of our microbiome than we did just 5-10 years ago, and what is clearly evident is that microbiome and health is really connected. And it also is pretty clear that the foundation is really laid down in the first years of life, where we see this, hopefully this drive of having bifidobacteria as the main gut family or bacteria family in the gut, and also that it can have long-term consequences if we do not get this in early life. But even throughout lifetime, we will meet challenges because the way we live, what we eat, we are stressed, we get antibiotics, we get medicine, and so we will constantly be challenged and have a dysbiosis in the gut, and this is where probiotics could also be one way to help to combat some of these dysbiosis, or help the commensal bacteria to do their functionality. And because what we also know is, of course, that this is really a complex ecosystem. So for one thing, it's a big surface area, but also that most of the bacteria that we have in the gut, they live in actually, or that we have in humans, are living in the gut, so 97%, and we have 5,000 species. This is the numbers that we work with today. So this, of course, is a huge ecosystem that needs to interact with each other, but they also interact and signal to the neurons. So we know a lot more now also on the gut-brain area. And then we have about 70 to 80% of the body's immune cells in there. So maybe they're not surprising that probiotics can actually also have a direct link or relation to the immune system. And what you can see to the right is just a cartoon kind of thing, or snapshot cross section of the gut. So the cells, the mucus, and the underlying immune cells. So this is where we will see that the probiotics will go down and interact with the immune system. The challenge then with working with probiotics and documenting the effect in relation to the immune system is, of course, that we do not have a single molecule that we can measure or target, but we are again facing this with a bacteria that goes into the gut where you already have a lot of bacteria, and we more than rely on doing real life studies where we can look into the clinical benefits, like or related to like the common cold, so the duration or the severity, because this will then be an indication of an effect on the on normal immune response. But we also have a chance to work with vaccine models. This is something that has been recommended some years ago, because then you actually challenge the immune system and you know that you will have a response elicited. So the response observed from the probiotics would then be expected to translate into a defense against other viruses like the common cold. Because so it's more a mimicking of a normal situation, because just taking blood samples would not really help us in really knowing if this would translate into benefits. So just to study immune markers would only maybe help us to tell us about potential mechanisms behind the clinical effects we see. So we prefer to look into real life studies or using vaccine models, and I will just show you some examples on these data that we have. And I will show you from two strains, as I mentioned at the beginning. It's two lactobacillus strain. One is the Lactobacillus rhamnosus LTG, and the other one is a Paracasei subspecies Casei LKSI 431. So one of the studies is a study in children attending daycare, where the risk of getting an infection, respiratory tract infection, gastrointestinal infection is 2 to 3 times higher than if children were not going to a daycare. And this was a randomized double-blind controlled trial with about 280 children. And they were then receiving a fermented milk drink for 3 months with 1 billion colony-forming units per day. And the primary outcomes were the number of children with gastrointestinal infections and respiratory tract infections. And the results you can see first on the top there, the primary outcome, you can see almost significant effect on the gastrointestinal infections, but on the respiratory tract infections, there was a highly significant effect with 43% in the LTG group who had a respiratory tract infection, whereas it was 67% of the children in the placebo group who had a respiratory tract infection. So really great results, so about 38% point in difference. Looking more into details of the data, it was clearly driven by upper respiratory tract infections with high significance, not on the lower respiratory tract infections. We can also see that the infections lasting longer than 3 days were significantly reduced. So this indicates that the severity was improved by LTG compared to placebo, and also the total duration was reduced, and the absence from daycare due to infection was also significantly reduced. So really strong evidence that really shows how a strain like LTG can benefit in a case where you are at risk of getting a respiratory tract infection. A similar study was actually by the same research group in Croatia, conducted also a study in hospital, so looking at nosocomial infections, so where the infections, of course, or the children are at risk of getting an infection when they are hospitalized. And also here they had significant results on both gastrointestinal tract infections and respiratory tract infections, looking at the risk ratio where are highly significant, and looking at the secondary outcomes at vomiting, the duration of both the gastrointestinal infections and respiratory infections, also at the longer end, showing that the severity was also improved in this hospital setting. So very strong evidence on the LTG strain in relation to the immune function. There are, of course, a lot of other publications in this area, but they all show a consistent relationship between the intake of the LTG strain and the body's natural defense, looking at different settings. So the two on respiratory health in daycare and hospital, another one in daycare, and then 2 studies that has used the vaccine challenge model, where they also show that there are effects in those studies. Moving then to just show some examples of using this vaccine challenge, and moving then to another strain, the LKSI 431 strain, where 4 publications have used, or 4 studies have used the vaccine model. 3 used the flu vaccine and 1 used the polio vaccine. So this was a study that we took under some years ago, a really large study with more than 1,100 subjects, randomized them to LKSA 431 or placebo. We had an intervention of 6 weeks with either 1 billion per day or placebo, and then a 9 weeks follow-up. And you can see at the study flow chart that after a run-in period, we started the intervention, and then after 3 weeks, we then injected with the flu vaccine, and then read out the response 3 weeks later. And during the whole period, we collected data on flu-like symptoms, work productivity, healthcare use, and quality of life. And the idea of providing the vaccine after some weeks of intervention, again, was to kind of mimic a real life, because we never know when we will get the common cold or the flu. So when we go to a season where we are at higher risk, of course we should take the probiotics, because this would be really the situation. So we are ready to combat any infection. What we found in this study was that we had a reduced duration of common cold symptoms in the LKSI 431 group. So in the placebo group, it was about 8 days, and that was reduced to about 5 days. So here we also found less use of healthcare resources and reduced number of antibiotic courses in the group. Similarly, a similar study was set up before that, a smaller study, same kind of intervention, same dosage. We injected the vaccine after 2 weeks and could here see a response on this vaccine-specific plasma IgG1 and IgG3, and a substantial increase in number of subjects obtaining this increase. So strong results, and also this was repeated in another study from 2017, and they found similar results and could look then into the seroconversion on the 2 out of the 3 strains in the vaccine, so the H1N1 strain and H3N2 strain, also demonstrating an effect of this LK43 strain. So these were sort of just a quick walk through some of the data that we have related to the immune system. And we just look into the potential mechanism of actions. We know that they can work at 3 levels. So in the lumen, so in connection to the mucus, where they can work on the dysbiosis in the gut. We know that they can compete with the pathogens for nutrients, so in that way sort of help exclude pathogens, but also stimulate the commensal bacteria in there. It can also help tightening the junctions in the gut, and also we know that they can secrete proteins that are important for infections. And reduce the permeability, and certainly they are directly and indirectly involved in stimulating dendritic cells and the Th1 response that we know is so important for fighting infections. So in summary, I would say that the emerging understanding of the microbiome health connection will certainly provide new insights for exploiting the potential of current and future probiotics, including the interaction with the immune system. And what will be important is, of course, to really understand the functionality, because that, of course, at the end of the day is the most important. And as I showed just now, is that 2 strains like LTG and the LKSI 431, they have high level documentation, high quality studies in immune area, and are certainly highly relevant today. And we also do have indication of what the potential mode of action could be of these 2 strains. So with that, I would like to thank you for your attention today.
Thank you, Dear Eskesen. Thank you for your excellent presentation. We took advantage from your lecture, and it seems that nobody have a question from you. Thank you, and we go to the third speakers. Thank you, Mr. Eskesen, for your presentation.
Thank you.
And the third speakers Dr. Saeed Mirdamadi. Dr. Mirdamadi, are you connected? Can you hear me?
Yes, yes, yes. But video, I think not connected. Camera.
Yes.
Can't start video.
You don't have your...
Okay.
Yes, okay. Hello, Dr. Mirdamadi.
Hello, thank you for your attention, and I must upload my slides.
Thank you. I hope you are well, Mr. Mirdamadi. As other speakers, also you have 20 minutes time and maybe 5 minutes for answering and question. Please start your lecture and let me introduce you, please.
Yes.
Dr. Saeed Mirdamadi, Iranian Research Organization for Science and Technology. And the lecture topic, Postpartum and Bioactive Peptides, the new horizon in farm food and safe biotherapy. Mr. Dr. Mirdamadi, please start your presentation. Thank you.
Yes. Do you see my slide or not? Okay. Do you see our slides?
Yes. Full screen and continue.
Yes. My lecture is Postbiotics and Bioactive Peptides, the new horizon in pharma food and safe biotechnology. This is my lab, functional food lab in biotechnology centers, and my colleagues works on bioactive peptides of lactic acid bacteria as a postbiotics and functional food. 2,500 years ago, Hippocrates said, let food be your medicine and medicine be your food. Food and health have always been closely linked together both. And medicine, foodstuff have the power to heal ability. Dietary intake of selected categories of food comprising active components regulated disease controlling mechanisms either as prophylactics or therapeutics. The main objective in the field of functional foods and health promoting bioactive compounds such as probiotics, prebiotics, phytochemicals, natural antioxidants, bioactive peptides. These foods are typically called as nutraceuticals, foodiceuticals, functional food, or medifoods. Probiotics have health benefit by modulating gut microbiome. Despite several health benefits of probiotics, they have some limitations, such as unknown molecular mechanism, strain-specific behavior, short-lived niche-specific action, developing antibiotic resistance, virulence gene transfer, difficult maintenance of viability and stability, ability to cause opportunistic infection, inflammatory response infection. The focus is gradually shifting from viable probiotic bacteria to non-viable, called paraprobiotics, probiotics-derived biomolecules, or postbiotics. Lactobacillus and bifidobacterium are the most studied probiotic genera and the safest microorganisms. So we focused on lactic acid bacteria in my laboratory. The functional food can be defined as any food that has a positive impact on an individual's health, physical performance, or state of mind. In addition of its nutritious value. So enhancement of biological defense mechanisms, prevention of specific disease, control of physical and mental disorders, slowing of the aging process are some regulatory effects in body process. The functional food divided to natural, transport, fortified, and enhanced foods. Some terms such as probiotics. Probiotics, ghost or inactivated probiotics, non-viable microbial cells, or crude cell extract, intact or broken, which when administered in adequate amounts confer benefit of the human animal consumers, orally or topically. Probiotics form are inactivated, dead, non-viable microbial cell or components of probiotic cells, such as teichoic acid, peptidoglycan, surface molecules such as PLIF, Fimbriya, polysaccharides like exopolysaccharides, EPS, cell surface associated proteins, cell wall bound biosurfactant. So, for inactivation of live bacteria, we can use from heat treatment, ultrasonication, high pressure homogenization, enzymatic hydrolysis, also autolysis, gamma use from gamma and ultraviolet, for disruption of cell and use these particles as paraprobiotics, and also assay the properties such as antioxidant activity and AC inhibitory. Heat-killed probiotic strain of lactobacillus showed antioxidant activity, anti-inflammatory effects, ability to support the inflammatory markers like IF6, TNF-alpha, and also enhance anti-inflammatory cytokines such as IL-10. Antagonistic effect against enteropathogenesis also was reported. The other terms, postbiotics. Postbiotics, non-viable bacterial products or metabolic products from microorganisms that have biological activity in the host. So, a non-viable bacteria, not component or dead cell, but fractions or metabolic products of microorganisms. Postbiotics are the complex mixture of healthy metabolic products, secreted components of probiotics in cell-free supernatants, such as enzyme, secreted proteins, short-chain fatty acids, vitamins, amino acids, bioactive peptides, bacteriocin, neurotransmitters, organic acid, biosurfactants, flavonoids, and also. Therefore, various postbiotics molecules, due to their non-chemical structure and non-specific mechanism, can inhibit some biological disorders such as inflammation, adhesion to pathogens to GIT, obesity, hypertension, coronary artery disease, anti-tumor activity, and oxidative stress. Postbiotics has advantages such as availability in their pure form, ease in production and storage, availability of production process for industrial scale-up, a specific mechanism of action, better interaction with cell receptors, enhanced interaction with the epithelial cell, and also probiotic characters such as gut functioning, evaluation of lactose tolerance, enhancement of immune function, antimicrobial activity, anti-biofilm formation, and some characters according to their specification. Postbiotics also has some advantages, such as no risk of bacterial translocation from the gut lumen to blood among immunocompromised subjects, no chances to transfer of antibiotic resistance genes, more natural to extract, easy standardization, transport, and storage, more stable, more controllable and safe. Risk and safety assessment is less complicated. The ideal natural functional food should fulfill the following criteria: low toxicity, stability of processing and storage, efficacy at low concentration, economic production, no side effect, metabolites of probiotics and lactic acid bacteria, especially lactic acid bacteria, and natural bioactive compounds of these strains have fulfilled this requirement and these advantages. An important note that regulation of probiotics, prebiotics, postbiotics. Japan is the first country to propose legislation for the specific regulatory approaches and in for food, and therefore several other countries have also structured this regulatory enforcement. FDA in USA, India, FSSAI, Food Safety and Standards Authority. In China, CFDA. In Europe, EFSA, European Food Safety Authority, and also other countries. In our lab, functional food lab in biotechnology department, we are studying on probiotic bacteria and their postbiotics, such as EPS, exopolysaccharide, bioactive peptide, bacteriocins, and other fermented byproducts. This lecture is an abstract of several research work and PhD theses in my research team in my lab. New peptides, first new peptides were purified, identified, and sequenced from fermented samples, lactic acid bacteria, yeast cells, and other probiotics. The peptides were assessed for biological characters such as antioxidant activity in different ways, DPPH and ABTS assay, ferric reducing activity power, FRAP, lipid peroxidation inhibition, protein oxidation inhibition, AC inhibitory activity, AC inhibition mechanism, anti-diabetic activity, and anti-aging activities. The proteins, precursor proteins, was digested by fermentation, lactic acid bacteria, or by enzymes. GI tract enzymes such as pepsin, chemotrypsin, or isolated purified enzyme of other sources such as protease of microorganisms. The proteins digestion to the bioactive peptides. Bioactive peptides usually contain 25 amino acid residues. These peptides have some activity, such as antimicrobial activity, antiviral activity, anti-cancer activity, immunomodulation, antioxidants, opioid effect, anti-osteoporosis, anti-inflammatory, anti-hypertensive, anti-thrombic, cholesterol reduction, anti-diabetic, and anti-aging activity. The yellow items were assessed in our researches. Around 10 new peptides were designed and chemically synthesized according to pattern of natural isolated peptides. Also, the structure function of original peptides and natural peptides analog were studied. On the other hand, as a risk assessment study, cytotoxicity, stability, side effects, mechanism, safety factors of new isolated and synthetic peptides were assessed. First of all, we focused on a fermented traditional camel milk we call chal, fermented camel milk, and a candidate for functional food, traditionally produced using spontaneous fermentation in Turkmen Sahra, Gulistan province in Iran. Microorganisms isolated from this sample and identified by molecular study and phenotypically. And also the bioactive components isolated, proteolytic activity of each strains assessed in fermenting cow and camel milks, and the best strains isolated and assayed as a production of fermented milk and assessed sensory characterization. After that, the character of antioxidant activity, AC inhibitory of best strain were studied. The comparative assessment of AC inhibitory and antioxidant activity of the best strain, Lactobacillus rhamnosus, PTCC 6037, were studied, and the best strain used for production of a safe functional food. For the best strain and safer strain, safety assessment of strains were done. Safety assessment in an important step to ensure the safety and quality of fermented products. Most of lactic acid bacteria species are included in the qualified presumption of safety list, but some lactobacillus strains involved in infectious disease such as bacteremia, urinary tract infection, and so on. For this aims, we tested more than 20 virulence gene in bacteria, identification of virulence gene, and also genetically or by phenotype study, antibiotic resistant, gelatinase activity, the carboxylase activity, hemolysin activity as a phenotypic study, and also antibiotic gene transferable antibiotic resistant gene and virulence genes studied genetically. Some strain were found to harbor at least 2 or more of the tested virulence genes, such as Enterococcus faecium strain, were positive several of these genes. So the best strain, the safest strains isolated for production of postbiotics, bioactive peptides, And novel beta-casein derived from this fermenting strains for antioxidant and AC inhibitory. These peptides identified, sequence, and by docking check the conformation and attachment to the target molecules. The second research in yeast cells, Chloromyces lactis and Saccharomyces cerevisiae, proteins extracted, purification of peptide, bioactive peptide were done, and by HPLC purified, by MALDI-TOF sequence, and after that chemically synthesize these peptides and also analog of natural peptides. And for safety assessments, toxicity in cockroach cells, protective effect against oxidative stress in cockroach cells, also expression of genes in the treated cells by our bioactive peptides, docking, AC inhibitory effect, antioxidant activity effect, anti-aging and anti-diabetic activity of these peptides were assessed. So we patented several sequence of peptides, around 10 peptides from Saccharomyces cerevisiae, and around 5 peptides from Chloromyces marxianus. All of them compact sequences, and each amino acid's position in the peptide and also effect of each amino acid were studied. After isolation, purification, and also design new peptides, each peptide purified and examined for biological activity from Saccharomyces cerevisiae, Chloromyces marxianus, and after sequencing, identified and checked for biological activities. These peptides are the safest peptides isolated from Saccharomyces cerevisiae, Chloromyces marxianus, with antioxidant activity, antihypertensive activity, and also the other peptides, 2 peptides we isolated, anti-diabetic activity. So production of antioxidant and AC inhibitory peptides from Chloromyces marxianus and also yeast Saccharomyces cerevisiae, purified, and function of these molecules and attachment to target was studied by bioinformatics and also in the laboratory in vitro assay. These peptides have several biological activity, and also each amino acid, character of each amino acid effect on the biological activity of peptides. A structure-function relationship of each peptide is so important to know how they act as a bioactive compounds. So all of experiments focus on safety, toxicity effects, and also terms of attachment in the target molecules. For safety assessments, we add H2O2 as a oxidant to the cell line, and effect of each peptides for protective effect against H2O2 was tested. These cells, these treated cells, examined as gene expression, effect of each peptide to expression of some genes, protective genes, also in toxicity to cell lines by flow cytometry, effect on apoptosis, necrosis, and also natural, safe, viable cells. In the final studies, we try to focus to production of a functional food, functional dairy food. So the best isolation bacteria used for production of functional fermented milk with all of this biological activity contain peptides and bacteria which have antioxidant activity, anti-hypertensive activity, anti-diabetic activity. I like to say that the future of food products, both nutritious character, maintenance and growth, and also pharmaceutics, health promoting activity, is the new horizon in pharma food and safe biotherapy. Thank you for your attention.
Thank you, Dr. Mirdamadi, for your excellent and valuable presentation. It seems that nobody have a question, and thank you for your presentation. Say goodbye to you. And we go to the 4 speakers. Dear Farzaneh Aziz Mohseni, and the lecture topic, a review lecture topic, biosafety assessment of probiotics. Dr. Aziz Mohseni, are you connected? Are you hear me? Dr. Aziz Mohseni,
yes.
Can you hear me?
Yes, I can hear you. You also have my voice?
Yes, we have your voice clearly. Hello to you, and I hope you are well. As other speakers, you have also have a 20 minutes time. Let me introduce you. Dr. Farzaneh Aziz Mohseni, Director of Persian Type Culture Collection, Iranian Research Organization for Science and Technology. Dr. Aziz Mohseni, please start your presentation. Thank you.
Just a moment, please.
Dr. Mirdamadi, please disconnect your sharing. Okay. Dr. Aziz Mohseni, please start it,
Yes.
full screen and start it. Thank you.
Hi everyone. At first, I should apologize for a misunderstanding when the colleague who contacted me for invitation. I asked him about the language of the presentation. He described me that the presentation could be performed in Persian, so I presented my lecture in Persian. Really sorry for this misunderstanding. Biosafety assessment of probiotics ارزیابی ایمنی زیستی
Excuse me,
پروبیوتیک.
Dr. Aziz Mohseni, excuse me. Would you please send your picture if you want? We don't have any picture of you. Would you please send your pictures, your film during the presentation? If you want, of course. Yes, thank you. Thank you. Continue. Excuse me. Please continue.
Okay. ارزیابی ایمنی زیستی پروبیوتیک ها مبحثی هستش که میخوام خدمتتون ارائه بدم. I think there is a problem. Okay. خب اجازه بدین از مقوله پروبیوتیک و معنای اون شروع بکنیم که خود کلمه پروبیوتیک به معنای برای زندگی هستش. اگه بخوایم یه سریعاً
Excuse me, dear doctor, please present in the English your presentation. Thank you.
I described for you at first that the colleague who contacted me, I asked him about the language of the presentation, and he said me in Persian. So I prepared my presentation in Persian, but the slide is in English.
And you can now present in English?
It's difficult for me. I am not ready for this presentation in English.
Yes, please present in Farsi.
Really sorry. I apologize everyone for this misunderstanding.
Your English very good, Dr. Aziz Mohseni.
Thank you. در سال 1906 آقای دکتر تیسیه اولین کسی بود که متوجه شد که و اشاره کرد که فلور روده کودکان مبتلا به اسهال با فلور روده کودکانی که سالم هستند خیلی متفاوت هست و تعداد زیادی از ارگانیسم هایی در مدفوع کودکان سالم وجود داره که در بیماران وجود نداره. در سال 1907 ایلی مشنیکوف برنده جایزه نوبل وابستگی بین غذای مصرفی و میکروبیوم روده رو به صورت یه فرضیه مطرح کرد و این فرضیه رو در واقع اینجور مطرح کرد که با تمهیداتی میشه تغییر داد فلور روده رو. در واقع تیسی و مچنیکوف اولین دانشمندانی بودن که متوجه شدن که فلور روده اهمیت داره و میتونه منجر به جایگزینی میکروارگانیسم‌های مفید به جای میکروارگانیسم‌های مضر با مصرف مواد غذایی بشه. در سال 1965 اولین بار در واقع اصطلاح پروبیوتیک وارد ادبیات بیولوژیک ما شد و این مسئله رو لیلی و استیلول به کار بردن این اصطلاح رو برای اینکه بگن بعضی از میکروارگانیسم ها موجب افزایش رشد میکروارگانیسم های دیگه ای میشن. در سال 1989 اولین بار در واقع مفهوم جدید پروبیوتیک توسط فولر مطرح شد که گفت مکمل های غذایی که حاوی میکروب های زنده باشن با تغییر بالانس روده تاثیر مفیدی میتونن در حیوان میزبان ایجاد بکنن. و بالاخره در سال 1999 92 هاونار و بیوسنتیولد تعریفی تقریبا مشابه ارائه دادن و باکتری گفتن که باکتری‌های زنده‌ای که توسط حیوان یا انسان مورد استفاده قرار میگیره با تغییر فلور دستگاه گوارش میتونه اثرات سودمندی ایجاد بکنه. و بالاخره در سال 1998 تعریفی ایجاد شد که تقریبا همه الان به همین صورت تعریف رو به کار میبرن. اونم عبارت از اینه که پروبیوتیک ها میکروارگانیسم‌های زنده‌ای هستن که وقتی به میزان کافی مصرف بشن موجب بهبود سلامتی میشن و تمام این تعاریفی که الان ما ارائه کردیم حاوی در واقع ویژگی‌های مشترکی هستن. این ویژگی‌های مشترک به صورت خلاصه عبارتن این که اولا پروبیوتیک باید میکروارگانیسم‌های زنده رو در بر بگیره. دوم این که باید دوز کافی از و مقدار کافی از اون میکروارگانیسم وارد بدن بشه. سوم که اثر مثبتی باید داشته باشه روی سلامتی. چهارم این که در واقع قدرت زندمانی و رشد مناسبی رو داشته باشه و بالاخره اثرات سودبخشی که در واقع اثرات سلامت بخش و مهمی که این میکروارگانیسم ها دارن اختصاصی استرین هست و اختصاص به گونه و جنس نداره، به این مفهوم که اگر یه استرینی ویژگی مثلا استرینی از لاکتوباسیلوس اسیدوفیلوس ویژگی پروبیوتیک داشته باشه دلیل نمیشه که تمام گونه های لاکتوباسیلوس اسیدوفیلوس این ویژگی رو داشته باشن و همچنین دلیل نمیشه که لاکتوباسیلوس ها کلا این ویژگی رو داشته باشن. پس این ویژگی اختصاصی استرین هستش. problem it seems that a problem I have encountered with my laptop. خب معیارهایی که برای اینکه میکروارگانیسم رو بتونیم در گروه پروبیوتیک بگنجونیم عبارتن از مقاومتی که در برابر شرایط اکستریم داخل دستگاه گوارش دارن، چه از نظر pH پایین و چه از نظر برخورد با موادی مثل املاح صفراوی، تواناییشون برای اتصال به سلول‌های اپیتلیال، تواناییشون برای کلونیزه شدن و استقرار در روده ها و جزئی از فلور روده شدن، یه ویژگی مهم نان پاتوژن بودن و توانایی تحمل این ویژگی دیگه خارج از ویژگی‌های بیولوژیک ارگانیسم است، توانایی در واقع تحمل در برابر فرآیندهایی که فارماسوتیکالی که منجر به تولید مواد پروبیوتیک میشه مثل لایوفیلیزیشن و بالاخره ویژگی مهم تکثیر سریع یا به نوعی جنریشن تایم کوتاه. در همین رابطه تستای این ویترو یا برونتنی که برای غربالگری سوریه‌های میکروبی برای اینکه ببینن توانایی بالقوه پروبیوتیک بودن رو دارن یا نه عبارتن از مقاومت در برابر اسید معده داشتن، مقاومت در برابر اسیدهای صفراوی داشتن، قدرت اتصال به رده‌های سلولی اپیتلیال رو داشتن، فعالیت ضد میکروبی بر علیه باکتری هایی که بالقوه میتونن بیماریزا باشن رو ارائه دادن، توانایی رقابت با میکروارگانیسم های پاتوژن از نظر چسبندگی به سلول های اپیتلیال و به این ترتیب باعث کاستن از میزان چسبندگی ارگانیسم های پاتوژن شدن و بالاخره دارا بودن فعالیت هیدرولازی نمک های صفراوی. تمام این ویژگی ها رو ما در استاندارد در واقع 19452 ایزیری میتونیم مشاهده و بررسی کنیم. خب اگه بخوایم یه نگاه کلی بندازیم روی گایدلاینی که WHO یا FAO در مورد ارزیابی پروبیوتیک ها ارائه کرده، میتونیم به این دیاگرام توجه بکنیم. ابتدای کار از قسمت بالا اگه بخوام خدمتتون بگم به صورت کلی، در واقع با شناسایی هر گونه ارزیابی پروبیوتیک با شناسایی ارگانیسم شروع میشه که این شناسایی هم ویژگی‌های فنوتیپ رو در بر میگیره و هم ویژگی‌های ژنوتیپ و منجر به تعیین در واقع جنس و گونه و استرین ارگانیسم میشه. مرحله بعد مرحله هستش که نباید ندیده گرفته بشه و اون هم دیپازیت کردن استرین در مراکز نگهداری منابع زیستی یا کالچر کالکشن ها هستن. بعد از این مرحله در واقع سیفتی اسسمنت یا ارزیابی ایمنی به طریق این ویترو انجام میشه و همزمان در واقع ویژگی‌های فانکشنال ارگانیسم چه از طریق این ویترو و چه از طریق آزمون‌های درونتنی حیوانی صورت میگیره. فاز 2 در واقع فازی هستش که کلینیکال ترایال انسانی رو در بر میگیره و اگر که ادعا یا کلیم مربوط به دارو بودن پروبیوتیک مطرح باشه، فاز 3 هم در واقع باید وارد مراحل کلینیکال ترایل بشه و در نهایت در واقع به عنوان پروبیوتیک فود میشه عرضه کرد این محصول رو به مارکت. خب برمیگردیم روی این گایدلاین، حالا توضیح میدیم که به چه ترتیب در واقع پروبیوتیک ها باشون رفتار بشه برای اینکه از موقعی که جدا میشه تا موقعی که ادعا میشه بشه وارد مارکتش کرد. اولین مرحله در واقع آیدنتیفیکیشن بود یا تاکسونومی و تایپینگ دقیق. ارگانیسمی که تحت ارزیابیه واقعا باید بر اساس متدولوژی، یعنی این صحبتیه که از سال 2003 که در فاو مطرح شد همونجا مطرح کردن با توجه به اینکه تکنیک‌ها تغییر میکنه بر همین اساس هم تست ها هم باید بر اساس متدولوژی های آپگرید شده و به روز باشه. بنابراین اگر که مثلا گایدلاین فاو 2003 رو با افسایی که مربوط به در واقع اتحادیه اروپا هست و در سال 2018 منتشر شده مقایسه کنیم، میبینیم که خیلی تاکیدها بیشتر الان بر روی ملاحظات مولکولی، آنالیزهای مولکولی هست تا فنوتیپ. به طور مثال در واقع آیدنتیفیکیشن الان دیگه اگر قراره برای ارگانیسم انجام بشه هول ژنوم سکونسینگ هست که نه تنها تاکسونومی رو مشخص میکنه بلکه در واقع جایگاه و نقشه کلی ژنوم رو چه از نظر متابولیت هایی که تولید میکنه و چه از نظر ویژگی های مختلفی که باید تو پروبیوتیکا مورد ارزیابی قرار بگیره و مورد توجه قرار بگیره رو پوشش میده. ولی برای تاکسونومی سطوحی مانند استفاده از ژنهای خاص مثل 16S یا ژنهای هاوسکیپینگ که ژنهایی هستن که تولید کننده پروتئین های اصلی ارگانیسم هستن، یا در مورد مخمرها و قارچ‌ها ITS و 18S هم میشه به کار برد برای اینکه آیدنتیفیکیشن رو در واقع یا شناسایی رو به صورت دقیق مشخص کرد. توصیه میشه که شناسایی یا آیدنتیفیکیشن چون امروزه واقعا یه مبحث کاملا تخصصی هست توسط یک لابراتواری که اکسپرت هست تو زمینه تاکسونومی انجام بشه و معمولا این آزمایشگاه هایی که اکسپرت تاکسونومی دارن چه تو زمینه حالا آنالیز ها و چه تو زمینه بایو انفورماتیک مراکز نگهداری منابع زیستی یا BRC ها هستن. بنابراین توصیه میشه که ارگانیسم حتما دیپازیت بشه بعد از اینکه در واقع کلیم پروبیوتیک بودن مطرح شد عرضه بشه به BRC ها و BRC ها موظف هستن که اونها رو تحت کاندیشن های در واقع رستریکتد یا محدود که این محدودها که مطلع هستین دو قسمت یا سیف دیپازیت هست و یا پتنت دیپازیت هست به خاطر اینکه حقوق مالکیت فکری کاشف در واقع پروبیوتیک شرکت یا فرد حفظ بشه حتما به BRC ها تحویل داده بشه هم برای تاکسونومی دقیق و هم برای امکان مطالعات بعدی ولی در قسمت پابلیک دیپازیت در واقع امانت گذاری نشه و در قسمت سیف و پتنت امانت گذاری بشه. بعد از اینکه تاکسونومی انجام شد، مقوله های مربوط به سیفتی مطرح میشه که خیلی خیلی اهمیت داره، از این لحاظ که در واقع پروبیوتیک موجود زنده است که در تعامل قرار میگیره در یه فضای زنده مثل جی آی و با میکروارگانیسم های دیگه‌ای که قراره که با همدیگه در واقع یه سری تبادلات داشته باشن. بنابراین چون وارد بدن میشه و چون با ارگانیسم های دیگه و با در واقع سلول های میزبان چه حیوانی چه انسانی قراره مواجه بشه، یکی از مسائلی که باید روش کار بشه و ارزیابی بشه، آیا بالقوه توانایی ارگانیسم توانایی تولید متابولیت های در واقع مضر رو داره، آیا توانایی تولید انتروتوکسین ها به خصوص امروزه برای باسیلوس ها که خیلی وارد مقوله پروبیوتیک ها شدن مطرح میشه، انتروتوکسین تولید میکنه، ویرولانس فاکتورهایی مثل همولیزین یا در واقع هیدرولاز هایی که آسیب زننده هستن به سلول ها تولید میکنن یا نه. و مهمتر از اون که امروزه یک مسئله عمده جهانی هست، آیا قدرت انتقال مقاومت های آنتی باکتریال یا آنتی میکروبیال رو داره یا در واقع حساس هستش به این مسئله. خطر در واقع اینو در نظر داشته باشین، خطر استفاده از میکروارگانیسم هایی که ژن های مقاومت دارویی رو وارد چرخه غذایی میکنن و موجب انتقال ژن های مقاومت به جمعیت میکروبی مستقر در داخل روده میشن، همیشه باید مورد توجه قرار بگیره، به خاطر اینکه امروزه ایجاد سوپر باگ ها از مسئله های خیلی مهم بهداشت و درمان هستش که در تمام دنیا مطرح هستش. از نظر حساسیت یا مقاومت میکروبی یه توضیحی بدم خدمتتون که ما مقاومتی که در واقع در مورد میکروارگانیسم ها داریم به دو دسته اصلی تقسیمش میکنیم. یکی مقاومت اینترنسیک یا مقاومت در واقع درونی یا نچرال و یکی مقاومت اکتسابی. جالبه بدونید که مقاومت وقتی که اینترنسیک هست مقاومتی هستش که اختصاص به یه گونه داره و وقتی که در واقع گونه تکثیر پیدا میکنه این مقاومت انتقال پیدا میکنه از نسلی به نسل دیگه. یعنی این مقاومت ذاتی هست، یه ویژگی ساختاری در تمام جدایی‌های یه گونه وجود دارن که موجب میشه که یه تارگت مناسب برای آنتیبیوتیک خاص در سلول وجود نداشته باشه. و اینم بدونیم که در واقع این نوع مقاومت یا مقاومت درونی واقعا مشکل سیفتی نداره و از جمله پارامترهایی نیستش که باعث بشه که مسئله پروبیوتیک بودن ارگانیسم خدشه‌دار بشه. ولی وقتی که یه ارگانیسمی که به صورت در واقع معمول توی خانواده و جنس مطرح شده حساس هست به یه ماده ضد میکروبی ولی ناگهان نسلی پیدا میشه که مقاومت داره، میشه مقاومت اکتسابی. مقاومت اکتسابی خودش به دو دسته تقسیم میشه، یا مقاومتی که به صورت موتیشن در اثر موتاسیونی که در تارگت در واقع آنتیبیوتیک ایجاد میشه رخ میده و باز هم این هم سیف مطرح میشه، ولی مقاومتی که توسط در واقع عامل اگزوژن وارد ارگانیسم شده و قابلیت انتقال داره، این مقاومتی هستش که در واقع میتونه خطرناک بشه و موجب میشه که در همین مرحله ارگانیسم از پروبیوتیک بودن خارج بشه. بنابراین اگر بخوایم از نظر فنوتیپیک این مسئله آنتی میکروبیال رو که اینقدر های اهمیت هست مورد در واقع بررسی قرار بدیم و تعریفش بکنیم، بهترین روش قدیمی تعیین MIC هست. برای هر ارگانیسمی بستگی بر مطالعات اپیدمیولوژیکی شده و در واقع آنچه که منتشر شده کاتاف ولیوهایی تعریف شده که بهترین در واقع جدولی که شما میتونید پیدا کنید در مورد این کاتاف ولیوها افسایی هستش که در سال 2018 منتشر شده و کاتاف ولیوها اونجا تعیین شده که مشخص میکنه که در چه حالتی ما به ارگانیسم میگیم حساسه و در چه حالتی میگیم مقاومه. در واقع اگر MIC از کاتاف ولیو پایین تر باشه ارگانیسم حساسه و اگر که بیشتر باشه مقاوم تعریف میشه. و باز امروزه همونطور که گفتم مهمترین مسئله WGS یا هول ژنوم سکونسینگ هستش که به راحتی ژن‌هایی رو که در واقع ایجاد مقاومت میکنن مشخص میکنه و از روی اون ژن‌ها خیلی راحت‌تر میشه ارزیابی کرد که این مقاومت اولا اکتسابیه یا نه و ثانیاً اگر اکتسابی هست از کدوم نوع هست. این جدول در واقع افسای 2018 هستش که کاتاف ولیو ها رو مشخص میکنه و هر دسته از میکروارگانیسم هایی که باید با چه نوع در واقع چالش آنتیبیوتیک قرار بگیرن تا ارزیابی مقاومتشون صورت بگیره. اینجا لازمه که یه توضیح خدمتتون بدم. MIC در واقع همونطور که میبینید پروبیوتیک ها الان دسته های مختلف از میکروارگانیسم ها هستن. بستگی به اون دسته نوع در واقع کاندیشن MIC با هم دیگه متفاوت هست. ارگانیسم هایی هستن مثل باسیلوس ها و یا انتروکوکوس که حالا باز براتون بیشتر توضیح میدم چرا وارد اینا میتونن وارد پروبیوتیک دسته پروبیوتیک ها قرار بگیرن. اینها رو میشه از روش در واقع CLSI و مولر هینتون به صورت معمول استفاده کرد، ولی برای لاکتیک اسید باکتری ها در واقع آنچه که معیار هست، معیار سنجش هست، روشی هستش که در سال 2005 توسط کلر مطرح شد و استفاده از محیط کشتی هست به نام لب ساسپکتیبیلیتی تست مدیوم یا LSM و برای این ارگانیسم های در واقع لب حتما باید از این محیط استفاده بشه. خب اگه بخوایم خلاصه کنیم صحبت مربوط اهمیت مقوله خیلی مهم مقاومت رو مشاهده میکنیم که هم در واقع تعیین کوانتیتیو MIC هست. اگر MIC از یک کاتاف بالاتر باشه در واقع مقاوم میشه، پایینتر باشه اکسپتبل رد میکنه. اگر که مقاوم باشه بررسی میشه از طریق هول ژنوم سکونسینگ که آیا اینترنسینگ پذیرفته میشه. اگر که در واقع در اثر موتیشن هستش این مقاومت اکتسابی باز هم قابل قبول هست، ولی اگر که ژن‌های اضافه شده باید در واقع از کاتگوری خارج بشه. دیگه از پارامترهایی که برای در واقع سیف اسسمنت بعد مورد ارزیابی قرار بگیره در واقع توانایی تولید توکسین و پاتوژنسیتی هستش که یکی از مهمترین مقوله هایی که تو این زمینه به کمک میکنه هیستوری در واقع استرین هستش، از چه کاتگوری از چه اکوسیستمی جدا شده و لیترچری که وجود داره در مورد این دسته و این خانواده چقدر در مورد توکسین زایی یا پاتوژنسیتیش میشه توش مطلب پیدا کرد. بهترین رفرنس در این مورد در واقع که میشه در موردش بهش استناد کرد باز افسای 2013 هست که اونجا بر اساس کرایتری هایی ارگانیسم هایی رو که سیف هستن یا تحت عنوان ارگانیسم های گراس جنرالی ریکگنایزد از سیف این ارگانیسم ها رو در واقع معرفی کرده که اگر تو اون کاتگوری قرار بگیرن خود به خود مسیر خیلی راحت تر میشه، چون بعضی از آزمایشات حیوانی و انسانی اصلا حذف میشه. بعد از اینکه در واقع ارگانیسم هایی که تو این لیست قرار ندارن، گراس یا لیست در واقع Qualified Presumption Safe یا QPS قرار ندارن، اون وقت در مورد اینها حتما باید ارزیابی ژن هایی که مربوط به ویرولانس فاکتور هستن ارزیابی بشه و از طریق هول ژنوم سکونسینگ و بعد مقایسه بشه با دیتابیس هایی که الان در سطح جهانی هست، مثل دیتابیس هایی که به صورت آنلاین میتونید دسترسی داشته باشیم مثل VSDB یا PAI دیتابیس و این در واقع مشخص میکنه که آیا توکسین، اینویژن فاکتور تمام از طریق در واقع مقایسه ژنومی هستش وجود داره یا وجود نداره که خود به خود اگه وجود داشته باشه باز حذف میشه. و بهترین مثالی که در این مورد میشه زد در واقع سویه‌ای مثل انتروکوکوس فاسیوم هستش که ابتدا چون دو دسته هستن انتروکوکوها، یک گروهی هستش که جنرالی توی محیط هستن و ویرولانس فاکتور ندارن، حساس هستن به آمپیسیلین، اینها رو الان امروزه مشکلی نمیبینن در اینکه اضافه بشه به پروبیوتیکا. و دسته دوم اونایی هستن که عفونت‌های بیمارستانی ایجاد میکنن. برعکس این دسته از ویژگی‌هایی که دارن اولا مقاوم هستن با آمپیسیلین و ثانیاً ژن‌های ویرولانس فاکتوری که مثل تولید کننده هیدرولاز هستن و واجد کپسول هستن این ژن ها، وجود این ژن ها نشون میده که رد میشه از نظر اینکه صلاحیت استفاده در این کاتگوری رو داشته باشه. برای اینکه یه مقایسه‌ای داشته باشن با اینکه ببینیم که امروزه چقدر کار ژنومی شده بیشتر تا اینکه این ارزیابی‌ها به صورت فنوتیپ صورت بگیره، در سال 2003 اسسمنت هایی که تست هایی که برای اسسمنت کارگروه WHO FAO پیشنهاد کردن همون پترن آنتی میکروبیال هستش، مقاومت ضد مواد میکروبی هستش که توسط MIC در واقع ارزیابی میشد. اسسمنت وجود فعالیت‌های متابولیکی هستش که اثر سو داره، مثل تولید همولیزین یا تولید لاکتات که میتونه اثرات نورولوژیک ایجاد بکنه. باز به همین ترتیب ساید افکت هایی که از روی مطالعات انسانی صورت میگیره، بررسی های پست مارکت که به خاطر اینکه تداوم داشته باشه بتونن این در واقع ارزیابی ها رو صورت بدن، در صورتی که الان وقتی که مسئله ژنتیکی اومده همه اینها ابتدای تو فازهای خیلی جلوتر مشخص میشه. و آیا این که مثلا خود ارگانیسم جزء خانواده‌ای هستش که توکسین پرودوسر باشه یا نه. حالا الان بیشتر اون
Excuse me, I interrupt you. You don't have any time. Please finish soon.
This is the last slide.
Thank you. Sooner. Thank you.
در کنار در واقع مطالعات این ویترو یا درون تنی، باید مطالعات مربوط به برون تنی صورت بگیره که اولین مرحله مدل های استفاده از مدل های حیوانی هست و در مراحل بعد مدل های انسانی مورد استفاده قرار میگیره که وارد فاز 2 در واقع ارزیابی میشیم، کلینیکال ترایل ارزیابی فاز 2 که در این مرحله در واقع دو گروه که انسانی، گروه کنترل که فقط کریر رو مصرف میکنن و گروه در واقع آزمون که کریر به اضافه پروبیوتیک رو مصرف میکنن، باید سامپل سایز مناسب باشه تا بتونن اطلاعات آماری رو در واقع ازش استخراج بکنن و افیشنسی رو به دست بیارن. و اگر که کلیم مربوط به این باشه که اثرات دارو درمانی داره پروبیوتیک، اون وقت وارد فاز 3 میشن که باید مقایسه صورت بگیره بین اثرات در واقع پروبیوتیک با تریتمنت استاندارد یا درمانی که به صورت استاندارد برای شرایط خاص صورت میگیره. باز به صورت خلاصه تمام صحبت هایی که کردم تو این در واقع سیستماتیک اپروچ رو شما میبینید که باید هم این ویترو چه تستایی صورت میگیره، این ویوو چه تستایی صورت میگیره در فازهای مختلف. در واقع از فاز 2 به بعد کلینیکال ترایل میشه و من باز هم عذرخواهی میکنم بابت در واقع ارائه به زبان فارسی، اطلاعی نداشتم و تشکر میکنم. Thank you for attention and thank you for your time.
Thank you, dear Dr. Aziz Mohseni, for your nice presentation. And it seems that nobody have a question from your lectures. Thank you. Goodbye, Dr. Aziz Mohseni. If Dr. Diarfu, if you are still with us and connected, and dear Eskessen, if you are connected, some participants have a question from you. Are you hearing me? Are you connected yet? Dr. Fu and Dr. Eskessen. Dr. Fu, dear Dr. Fu, can you hear me? Dear Eskessen, are you connected yet? Some participants have a question from your lectures. Oh, excuse me, it seems that's not connected. And during the other speakers, we can connect to dear Eskessen and dear Dr. Fu for the answering the question of participants. And for the last one, Dr. Jiefu, Dr. Eskesen, are you connected yet? If you are still with us and you are connected, confirm us for the some question that have presentation want to answer you. And continue the program with the last presentation. The last presentation is about the stability of probiotic bacteria in dairy products. Let me introduce myself. I'm Dr. Mardan Furubi, Manager of Research and Development Department of Iran Dairy Industries, Pegah Company, Esfahan Pegah Company. Let's start with my presentation. In the name of God, stability of probiotics bacteria in dairy production process. Probiotic foods. A probiotic of live microorganisms which when administered in adequate amount confer a health benefit on the host. In the recent years, there have been an increasing interest in probiotic foods which has stimulated innovation and fuels the development of new products around the world. Probiotic bacteria have increasingly been incorporated in food in order to improve gut health by maintaining the microbial gastrointestinal balance. The most popular probiotic foods are produced in dairy industry. And that's question, why in the dairy? Because the fermented dairy products have been shown to the most efficient delivery vehicle for live probiotic to date. In this paper, in this review paper, we will discuss the application of probiotic microorganisms in dairy products and the stability in processing. The purpose of this review evaluated the resistance or viability of different probiotic bacteria under several conditions, such as the processing, food composition, storage, freezing, thawing, refrigerator, temperature, oxygen, pH, gastrointestinal environment, and packaging. The factor 1, effects of processing, storage, and food composition on probiotic bacteria viability. It's crucial that the food containing probiotic maintain the number of bacteria over than 7 CFU per gram until the end of the shelf life, as there are several environmental factors that can affect the probiotic, such as the acidity, oxygen stress, storage temperature, co-culture competitor, the pressure, moisture content, and etc. Each factor that affects on a viability or stability of probiotic during the process. For example, in processing of dairy or every food and product in the line, some process factor and some process parameter affects on the stability and viability of probiotic during the processing. For example, in dairy products, in dairy company, during the pasteurization, during the transfer the ingredient, and during the every kind of sharing that affects on the ingredients, maybe affects on the account of the probiotic. Processing parameters like a heat treatment, incubation temperature, cooling rate of products, packaging materials, and storage method scale of production. For example, microencapsulation of probiotic in different polysaccharides was proven to be an ideal way to preserve and protect the cell from detrimental factor during processing. storage or resistance in the gastrointestinal transit, as many studies demonstrate it. Another factor we discussed about is food parameters, the parameters that belong to the ingredient or every food that used in the processing line. For example, food parameters like pH, titratable acidity, molecular oxygen, water activity, percent of salt, sugar, and chemical like hydrogen peroxide, bactericide, artificial flavoring, and coloring agent. And some studies show that the nutritional value of the food affected on the viability and stability of probiotic bacteria. For example, the value in carbohydrate, salt, mineral, dietary fiber, vitamins, fatty acid, amino acid, and protein calculated that increase the viability of bifidobacterium and others and other probiotics. For example, if you have a soy, skim milk, or inulin in your formulation, for example, in formulation of yogurt, in formulation of ice cream, in formulation of some kind of cheeses, if you have some chemical, some nutritional substance like skim milk and inulin, you can improve your viability and establishment of your probiotic. It seems that as a role of probiotic. And the other factor, microbiological parameters, strain of probiotic, rate of proportion, and proportion of incubation. Example of probiotic bacteria using probiotic dairy products. Normally, this kind of bacteria using the probiotic dairy product like lactobacillus species, this kind of species that belong, bifidobacterium species, and other species like Streptococcus, Thermophilus, Propionibacterium, Yersinia, and other species. And the other factor, effect of temperature and oxygen on probiotic bacteria viability. Fermentation temperature is crucial for the viability of probiotics. The favorable temperature must be between 37 up to 43 degrees centigrade, but more than the 45 degrees centigrade will destroy the fraction of bacteria and population. And as you know, oxygen is another toxic factor for the several of probiotics. So scientific discovered method to lower oxygen level, for example, the fermentation under vacuum or intelligent packaging like the multi-layer with oxygen scavengers in the material. The continuous exposure to oxygen under acidic condition during storage is the main reason for the reduction in probiotic count. The other factor, effects of freezing, thawing, and refrigerated condition on probiotic bacteria viability. Freezing and thawing are affected the probiotic as the integrity of the cell membrane is damaged during the freezing process, so metabolic life can be stopped. Additionally, during thawing process, the microbial cell can attack the probiotic due to osmotic effect. A study shows that some of species like Lactobacillus johnsoni and Lactobacillus rhamnosus show high survival rate in retail manufactured ice cream with no decrease in the population inoculated in Italy. During storage, up to 8 months for Lactobacillus johnsoni and 1 year even for Lactobacillus rhamnosus. Viability of different probiotic bacteria during exposure to simulated gastrointestinal condition. In order to obtain a survival probiotic bacteria under gastrointestinal condition, and encapsulation method is required. Encapsulation method maybe with a microencapsulation, maybe with an arginate and chitosan coating, improve the survival during the exposure to adverse condition of the gastrointestinal tract. And some studies show that demonstrated that it's an interesting text that I read before. Some studies demonstrate that a little chocolate can improve the viability of the probiotic during the gastrointestinal environment. Some studies demonstrate that the chocolate was an ideal matrix for protecting the probiotic cell during shelf storage and for enhancing their property to resist over the gastrointestinal tract environment. And other factor that affects on the viability of the probiotic bacteria, effects of different packaging on probiotic bacteria viability. A positive effect of the viability of probiotic culture have been attributed to food packaging because there are generally the probiotic bacteria, generally anaerobic or microaerophilic bacteria. Thus, the glass packaging, due to its low oxygen permeability, favored for the survival of probiotic culture. The polyethylene, in compare of glass packaging, we compare two kind of packaging which each other. The polyethylene packaging, in compare of glass packaging, is permeable to gas and allow diffusion of oxygen into the product during the storage and reduce the shelf life of the viability of probiotic. Some studies show that conclude that the glass packaging was more appropriate than the plastic packaging in maintaining the viability of probiotic culture, but it's so expensive that more than the polyethylene, with no effects of packaging on the physicochemical characteristics even. A conclusion. In order to improve the viability of probiotic microorganisms during food production, microencapsulation is shown to be the key process. There is a general interest in improvement of physical and mechanical stability of the polymers using probiotic encapsulation. As I told, like as a chitosan, like as a sodium alginate and other substances. To ensure high population of probiotic not only in food during the storage but also after gastrointestinal digestion, or bacteria or probiotic bacteria may be safe during the gastrointestinal environment or during the shelf life in refrigerator or in ordinary environments. The carrier plays a very important role and should be carefully examined, considering the main constraints for the survival of probiotic: low oxygen tolerance, low acidity tolerance, and the convenience of the addition of prebiotics for or growth promoting factor in the formulation of probiotic food. It's crucial to be aware of probiotic strain technological properties. They are usually selected based on this aspect. Also, when a mix of probiotic bacteria is chosen, always the attention always should be kept in the mind and the antagonist relationship be considered. Thank you for your attention, dears. Does anybody have a question? Excuse me, dear SKSN or dear cooling food, you don't connect it? Are you hearing me for the answering some question?
Sorry. Yes. Sorry,
Yes.
I just come back. Yeah.
Thank you, dear Dr. Cooling food. Would you please connect your picture?
Yeah, I was. Yeah.
Thank you.
Okay.
Thank you. Hello again, dear. I hope
Yeah.
you are well. And some of my colleague wants to thanks to you and have a question. Let me connect my colleague, Dr. Faizabadi, the master of the University of Tehran Medical Science, and my colleague, Mr. Faizabadi, wants to thank you for you and have a question from you and from Mr. Eskesen. Mr. Faizabadi, please turn on your microphone. Dr. Faizabadi, we have your picture, but yes, thank you. Please. Dr. Faizabadi, we don't hear you. We can't hear your voice.
You have my voice now?
Yes.
Okay.
Clearly.
Okay. Hello, Professor Fu. Hello, Professor Miramadi, and all other invited presenters. I had some questions, but before this, I would like to thank you, all the presenters, and for the very informative data and information to give us. Because of the pandemic of COVID-19, I would like to take the opportunity to ask questions from the speakers, the first speakers, the second one, and also Professor Miramadi about the importance of the probiotics or postbiotic products to inhibit the growth and spreading of viruses such as COVID-19. I remember that Professor Mirdamadi mentioned about some peptides who discovered at their labs that showed antiviral effects. I appreciate if you could some explanation about the future usage of these peptides or postbiotics on the antiviral effect. Thank you.
Okay, thank you very much for the question. Actually, it is a very, how to say that, hot topic about the COVID-19 pandemic, effect. And actually, in order to answer your question, I haven't done anything on, coronavirus, because this one you need clinical trials, and we don't have the facility to do it in my UC, and only certain laboratory in the Ministry of Health that can perform that kind of activity. But to your information, actually, some of the probiotic have been reported to have impact on the COVID-19 patient. That is probiotic. But postbiotic, so far, I don't think anybody has done that part of the work yet. Actually, I'm very keen to try my postbiotic on those COVID-19 patients if possible. But that one, we need help from the medical doctor to try to, try out on the postbiotic product. Because our postbiotic product, if you try to Google through our publication, we had done a lot of work on the postbiotic that we developed since year 2002 until now, right? And we have been testified or verified by using various animal models, and they have broad health impact. And they do not specific to one aspect of health, but the postbiotic actually, through our knowledge, to our understanding, they help the overall health condition of the host. Means that they start to install or restore the gut health first. From there on, once when the gut is healthy, the overall health will be restored by itself. So the immune response I showed you just now, the IgA, IgM, and IgG, IgA, and IgM actually can be induced automatically by feeding the postbiotic. But of course, different postbiotic, they have slightly different effect. There's no doubt. Yeah. But all of them, they can elucidate the overall health impact on the host, regardless what is the health status of the host actually. So actually our intention here in Malaysia or in my research team, we try to use postbiotic as preventive measure. Yeah, to induce or to restore or to try to encourage the good health condition of individuals. By having the good health status, it will help the individual to fight or to against any infections, including the virus we believe, and including the bacterial pathogens as well. So this is what we are trying to test on the human species now. We have been testing on the various animal models, like I said, that rats, lambs, chickens, pigs models, and also the fish as well. All of them, they have the same trend of the positive health impact. Of course, there's slightly different level of the health impact, yeah, but they all show the same positive health impact in various types of animal models. So we are very strongly believe the postbiotic actually is more safer than probiotic to any host or any patient that required, yeah, the restoration of the health after certain infection, including the cancer patient as well. This is what we are targeting on later on. I hope that I answered your question. Okay.
Thank you very much. About probiotics, as you know, in these days, one of the clinical symptoms of COVID-19 is gastroenteritis. And at our hospital, some of the infectious disease specialists usually prescribe some probiotics in the market to the patients. And you mentioned that you have tried this on the patients. Could you please tell us about the results and the background of the patients?
Sorry, actually, the kind of clinical trials we need, the help from the medical doctors to run that. It is a clinical trials actually. Yeah, so I think to our knowledge, before you can embark to the human clinical trials, we have to test on the animal models first, right? For the first two barriers, normally you use one species. After that, you need to use more than one species to counterproof it before you can start to feed to the human being. I think the first two barriers we have gone through. Yeah, we have a lot of publication, actually more than 20 now in hand, to show that postbiotics are really safe, yeah, to be used as a supplement now. But in order to do clinical trials, we really need a lot of support from the medical doctor to do it. And this is what we are thinking of doing that. I don't mind actually to send a product to Iran. If you think that you all can try out, I don't mind to send. We need to have some collaboration work with you all on that medical aspect. Actually, they also can reduce the blood cholesterol as well. So this is very, very interesting, which we didn't expect this thing happen as well. So they behave like probiotic cell without the living cell here. And because of that, without living cell, you can apply it to any patient, even though immunocompromised patient, because they won't cause bacteremia side effect. So this is a very, very important criteria. And I think probiotic sometimes is not meant for infant as well. So those immune response is quite low, or immune response status is quite weak. Normally, the probiotic lifestyle is not recommended, if not mistaken, right? So under that scenario, postbiotic is safer to be, used as a supplement to help those individual need to restore their health condition. And we actually recommend to use it as something like vitamin types of supplement daily in order to improve the health condition for the preventive measure, preventions, reaction. And this is what we actually targeting, try to encourage that kind of, supplement. So yeah, I hope that, I answered to your question there.
Thank you very much. May I ask Professor Mirdamadi same questions too, if there is any time? Professor Mirdamadi? Okay, he's not on, I think. Thank you very much. for
Hi.
participating.
Hello.
دکتر میرآمدی؟
Yes.
سلام
Do you hear
احوال شما؟
me? حالتون
Yes.
چطور؟
Alhamdulillah. Thank you very much. I would like to thank you for coming to this webinar, and from your presentations. I put the questions and typed there about the benefits of these products that you develop in your laboratory against bacterial pathogens, and also about the viruses. Have you any program to check these products against COVID-19, and how you can see about the future usage of these products against viral diseases such as COVID-19?
No. We have enough facility for COVID-19. We haven't any facility. So we test our products against bacterial pathogens such as E. coli, Listeria monocytogenes, Bacillus cereus, and some molds.
What about co-infection between the viral disease such as COVID-19 with E. coli and also other bacterial pathogens? Do you think that removing such pathogens by these postbiotics or probiotics could improve the condition of the patients?
Sorry, the sound is not, quality of sound is not good.
Yeah. About the using of these biological important products against bacterial that co-infected with viral diseases, I ask. Because in those animals, as Dr. Fu mentioned, we have problems sometimes in chickens or farm animals that use probiotics as a growth promoter, for example. But the advantage of some probiotics is to remove or reduce the number of bacterial pathogens, for example. In case of co-infection between viral infections and bacterial infections, do you think that such products can help to improve the condition of patients in animals or humans?
We use the bacterial strain probiotics and also some products such as bacteriocins in food as a food additive, and examine the food such as cheese for pathogen bacteria. They affect in pathogen bacteria in the food as a food additive and preservative, natural preservative. And also I saw some reported against viral pathogens, but I don't think that in food as a reservoir of viral infection maybe don't affect, because viral particle alive in the cell, in the host cell. And if I think about effective these products on viral, we think to disinfection or not biological activity, because a virus is not alive outside the cell.
Thank you very much.
Sorry, I don't hear you.
Thank you very much, Professor Nidamari. Thank you, Dr. Furu.
Thank you for your organizing this webinar. Bye.
I have no question, Dr. Furu. If other audiences, I leave the microphone to the colleagues, and I thank all the participants before leaving you. Just to inform you that we have soon a webinar on food microbiology next month, and also the second topics on probiotics again. So the Iranian Society of Microbiology will inform you about the future events. And I apologize for presentation. It was some problems and misunderstanding, but all the slides were in English. And I asked Dr. my colleagues, Mohseni again, to come to present the slides in our future program in English. It was very nice and informative. Thank you very much.
Thank you, dear. Thank you. Thank you, dear Dr. Fu, and thank you, dear Dr. Faizabadi, for your question. And thank you for all participants and speakers. I would like to ask you, all participants and speakers, to turn on your videos for take a photo for memory and group photos with each other. If possible, all of the participants, please turn on your video, and all of the speakers turn off your video to take a photo for the memory, please. And I especially thanks to all of the speakers, Dr. Mirdamadi, Dr. Aziz Mohseni. A special thanks to the international speakers, dear Dr. Fu and dear Eskeson staff disconnected. Thank you for your participate and for your excellent presentation and excellent lecture. I hope that you have a good time, and for the end of our program, please turn on your video to take a photo from all of you. Dear participants, please turn on your video for taking a photo. Now we have 100 participants, but nobody on their video. Please turn on your videos. Dear participants, okay, technical supporter, Mr. Alaidini, please took your photo with these 3 or 4 pictures. Mr. Alaidini, can you hear me?
Thank you very much, Dr. Fouroughi. We took a lot of photos with the participants. It was great, and thank you all the participants to stay with us until now. Thank you again, Dr. Faizabadi, Professor Fu, and again, Dr. Fouroughi, for the nice presentation, and also moderating, also Dr. Mirdamadi, and all of the lecturers, and a special thanks to Dr. Abdullahid for organizing this session. Thank you all.
Thank you very much. Thank you for your participation.
Thank you very much.
Thank you.
Thanks for everybody. Hope stay safe and stay healthy.
Thank you, dear Dr. Fu.
Thank
Have a
you.
good time. Yeah.
Bye. Yeah.
Bye. Goodbye. Goodbye.
Bye.
Bye.
Goodbye.
Biz